To introduce our guest speaker tonight, Cadet Ryan Kreiser. So I've had the pleasure of knowing him for the past couple of years when he joined our department, the Department of Chemistry and Life Science at the United States Military Academy. So I believe this is one of the first times where we've had a student serve as a guest speaker for our evening seminar or the ACS Mid-Hudson seminar. So I'm really proud of the caliber of research that he's done and the accomplishments, academic and professional accomplishments that he's achieved to date. So I think this is a great opportunity for him to share his research and for you to learn more about the type of research that we do at the United States Military Academy. And of course, to extend bridges, I mean, we participate quite prolifically in recent years at the Undergraduate Research Symposium. So that's been a great opportunity for our institutions to get to know each other better within the Mid-Hudson community. So this further reaches out and participate and allows us to share with you our resources and what we do, and then continue to build bridges and relationships. More about Ryan. So he is currently a junior at the United States Military Academy. He is a chemistry major. He is in his leadership role. So as some of you may know, the United States Military Academy is multifaceted in that they have the academic side, the military side, and the leadership side. And the athletic side. So he is absolutely well-rounded in those disciplines and those domains. He started doing research with Dr. Limbacher in November 2019. And since then, he's been working on looking at molecular countermeasures against protein misfoldings. So there's applications to that with neurodegenerative diseases such as Alzheimer's, but then there's also the military application of it with traumatic brain injury. So within their group, they have about 9 students and 4 faculty members, ranging from military and civilian. So it is quite a very prolific and robust research effort that Dr. Limbacher leads. And so it's been very rewarding to see their progress through the years. And then I am excited to hear, to have Ryan share their research with you. So Ryan's current research investigates the relationship between the lipid composition of cellular membranes and toxicity with the aim of ameliorating the toxicity of misfolded protein oligomers and folded toxins. So within his current work with Dr. Limbacher and the group, he's actually been a co-author on several publications and a first author on a review paper. I'm excited to have him share his work with you. He's also won the Undergraduate Research Symposium paper last year, so he was one of the recipients of that, and he is a recipient of the Stamps Scholarship. And so upon his, as he continues on to study in this discipline, he does aspire to pursue a PhD in biochemistry and then work at a national lab. And of course, at some point, he does have to serve in the Army, so he aspires to be a logistician. So logistics officer, doing great things, supporting our nation, and then also supporting from the academic side as well as an Army officer. So with that, I'll turn it over to Ryan so that he can share with you his talk on combating the cytotoxicity of misfolded protein oligomers in a neurodegenerative disease. All right. So.
Thank you, Colonel Wynn. Good evening, everybody. I can't really top that introduction, so I'm going to try. Let me just pull up my screen. Okay, so if I share it right now, should be all set. And everybody see this okay?
Yes.
All right. Good evening, everybody. Thank you all for coming out. I really appreciate you taking the time, and I look forward to sharing what I have. So as Colonel Wynn said, I'm Ryan Kreiser. I'm a junior at West Point right now. They call us cows for a reason that is older than me. And I've been working with Dr. Limbacher for the past 18 months or so. Here's a photo of the group that was taken last fall. And I'm going to be talking about the research that we're doing, the research that other researchers in the field of protein misfolding diseases are doing right now, and sort of organizing that for you and hopefully giving you an understanding of what the field looks like at present and what the directions forward are. So a little bit of the agenda for tonight. First, I'll give you an introduction to protein misfolding diseases, how they occur, what they are, what the sort of central mechanism of action is for those. And then I'll talk about some of the different mechanisms by which researchers are looking at taking care of protein misfolding diseases, trying to eliminate their toxicity to cells by altering the kinetics of oligomer assembly, targeting oligomer physicochemical properties, protecting the cell from oligomer interactions directly, making use of the body's own protein homeostasis system. And finally, I'll talk about the role of membrane lipids and my recent work on that. And I'll wrap it up and we'll have time for questions at the end. So protein misfolding diseases, like a bacterial infection or a viral infection, are a class of disease. They include a number of diseases. I'll talk a little bit about that in the next slide. The most common protein misfolding disease is Alzheimer's disease, which currently affects 52 million people worldwide. The incidence of Alzheimer's disease is projected to approximately triple by 2050. The second most common protein misfolding disease, Parkinson's disease, also affects 10 million people worldwide right now, and it's predicted to grow in incidence as well. So Alzheimer's disease, as you can see, affects the brain. It causes death to neurons in the brain through extracellular and intracellular protein aggregation. So the amyloid beta plaques you can see here in this sort of micro scale illustration are aggregates that occur out of the A beta 42 protein within cells, or excuse me, between cells. And within cells, there are neurofibrillary tangles caused by the aggregation of the tau protein. So protein misfolding diseases as a class are incurable. They're progressive, they get worse over time. And they're caused by the aggregation of misfolded protein in whatever tissue system that they're affecting. So the three main classes of protein misfolding diseases are neuropathic, so localized to the brain, localized non-neuropathic to any other part of the body, or systemic to multiple different tissue systems. So some notable examples are Alzheimer's disease, Parkinson's disease, other types of amyloidogenic dementia, Huntington's disease, there's amyloidogenic parts of type 2 diabetes. If you are older than me, you were probably around for the mad cow disease scare. Spongiform encephalitis is also a protein misfolding disease. And finally, chronic traumatic encephalopathy, CTE. There's more recent scholarship that suggests that's also a protein misfolding disease. CTE is of particular interest to the Department of Defense because it affects veterans who have been deployed overseas and have suffered repeated sort of concussive shocks to the brain. So how do protein misfolding diseases happen? Essentially what happens when your body is making a protein is the mRNA gets translated through the ribosome and it forms a peptide chain. So that polypeptide chain initially exists in an unfolded state. That unfolded state sort of exists in disorder until it reaches a lower energy state as it folds. So ideally, an unfolded protein will go from an unfolded state to an intermediate state to the native state. The native state is the correct shape of the protein. It performs the function that the protein is meant to perform. So for an example, hemoglobin, the native state is the state that's able to ferry oxygen around the bloodstream. However, proteins can also fold into the wrong shape. And when they fold into the wrong shape, they exist in a state that's not as low energy as the native state. In order to get to a more stable conformation, the protein can either unfold and refold into the native state, or in the case of protein misfolding diseases, aggregate misfolded proteins to begin to form protein misfolded aggregates. So when multiple misfolded proteins have aggregated together, they form oligomers, and those oligomers eventually add more and more monomers and form amyloid fibrils. The amyloid hypothesis is the most supported mechanism of action for protein misfolding diseases. And the amyloid hypothesis postulates that the aggregation of misfolded protein is the primary cause of symptoms in protein misfolding diseases, and that the oligomeric intermediate species of protein misfolding aggregation is the primary cause of cellular toxicity. So rather than the amyloid fibrils, which can grow to very large sizes to the point of being visible with the naked eye, the oligomers that comprise a couple misfolded proteins aggregated together interact with the cellular membrane and cause toxicity. The way this happens is that these oligomers are highly hydrophobic, and they interact with the cell membrane. They can interact with multiple different receptors in the cell membrane or with the membrane lipids themselves through a generic mechanism. And once they interact, they embed themselves in the membrane. They form holes in the cellular membrane, and those pores allow calcium 2+ ions to escape, which disrupts the ion homeostasis within the cell and eventually causes cell death as the cell's unable to perform the functions that are required, that require calcium. So the reason that protein misfolding diseases are progressive is because of something called the aggregation cascade. The aggregation cascade is essentially the kinetics of amyloid fibril formation. So the first thing that happens is there are a number of monomers. Those monomers exist in here. Let me pull up a laser pointer. I can highlight this better. So these monomers exist in their misfolded state, and they're sort of floating around in the area of interest. The way they begin aggregation is by interacting with each other randomly. This is called primary nucleation, and this is how the first oligomers are formed. As primary nucleation continues, eventually those oligomers aggregate into fibrils. Now, aggregation into a fibril is a big deal because it enables the second part of the aggregation cascade, which is secondary nucleation. In secondary nucleation, the sort of side of the amyloid fibril acts as a nucleation site for misfolded proteins to interact and aggregate, which increases the rate of oligomer and then fibril formation very significantly. So if you can see on the diagram, the rate of reaction here goes from 10 to the -4 to 10 to the 4. So that's that's 8 orders of magnitude faster than primary nucleation, because you're going from what's essentially random interactions of molecules to something that's more directed. And the more fibrils there are, the more that's able to happen. Excuse me. So ever since the amyloid hypothesis gained prominence about 30, 35 years ago, scientists have been working to sort of attack that mechanism of action in order to protect cells and eventually protect humans. So there are many potential therapeutics that researchers have tried over the past few decades, and there's several distinct lines of inquiry, which I will discuss right after this. So as I said in the agenda, these are generally modifying the kinetics of oligomer assembly, modifying the physicochemical properties of oligomers, protecting cell membranes from oligomers, and taking advantage of the protein homeostasis system. The first one of these that we'll talk about is altering the kinetics of oligomer assembly. So here in this concept figure, you can see some of the ways that oligomer assembly can be inhibited. This green curve represents the normal sort of rate of aggregation for monomeric protein into fibrils. So here you can see this relatively flat part is where primary nucleation is dominant. That's called the lag phase. As secondary nucleation kicks in, the rate of reaction increases, and the protein reaches the point at which half of it's aggregated. That's where the rate is at its highest. That's also what triggers the half-time measurements of protein aggregation. Then the protein continues to aggregate until it runs out of monomer, at which point aggregation levels off. So there are a few ways to address protein misfolded aggregation. The first one is to inhibit primary nucleation. So if you inhibit primary nucleation through some molecular countermeasure, you're making it more difficult for the molecules of misfolded protein to interact randomly and form oligomers to start. As you can see, this has the effect of increasing the lag phase, but once secondary nucleation becomes dominant, since you're not targeting secondary nucleation, it's no longer effective at stopping the rate of reaction. Another way to attempt to accomplish this is by targeting secondary nucleation, which doesn't have any effect on the lag phase, but does have an effect on the maximum rate of reaction and does increase the half time for the protein. Finally, you can employ a countermeasure that prevents elongation of fibrils. This is generally not done because preventing fibril elongation doesn't prevent secondary nucleation, which is the primary source of oligomers. And as we discussed earlier, oligomers are the toxic species. So preventing the elongation of fibrils doesn't directly reduce the amount of toxic species. There are many different molecules that people around the world and our group in specific have used to inhibit aggregation. Some good examples are many different types of antibodies, many polyphenols, flavonoid molecules. So two examples that we'll talk about further here are epigallocatechin gallate, which is found in green tea, and resveratrol. So these are two polyphenols here that have interesting effects on protein aggregation. EGCG in specific has the property that it's able to disaggregate fibrils into the monomer phase, which is significant because most of these molecules are only able to inhibit further aggregation rather than being able to get rid of aggregates altogether. So some work that our group has done over the past few years has been looking at rationally designed antibodies and their ability to interact with the A-beta-42 protein implicated in Alzheimer's disease. So these antibodies are rationally designed to bond to some specific residues in the A-beta-42 protein, and they can affect different steps in aggregation because of this. So for instance, this designed antibody, which binds to residues 18 through 24, as you can see, increasing the concentration here, this is the highest concentration, this red line, greatly increases the lag phase. Whereas this antibody, which bonds to residues 34 through 40, has the effect of slowing the half time. So in addition to molecules that slow down the rate of aggregation, another strategy is to enhance the rate of aggregation. This sounds counterintuitive, but the idea is as follows. By increasing the rate of aggregation, we can increase the rate of conversion from monomer to fibril, which means that any given oligomer spends less time as an oligomer and has less of a chance to be able to interact with cells. This is important because while oligomers are not stable, we say that they exist in a metastable state, which is to say that while each individual oligomer is relatively short-lived, the creation and destruction of oligomers happens at such a rate that the population of oligomers remains stable. So by increasing the rate of aggregation, it's possible to reduce the population of oligomers that are present in order to reduce that sort of toxic burden on cells. So one example of this is the aminosterol tridoscamine. This is a derivative of cholesterol that Dr. Limbacher has been studying since his PhD time in Cambridge, and that I've had the opportunity to study a little bit here at West Point. So as you can see, increasing the concentration of tridoscamine has the effect of reducing the half time for the protein aggregation and increasing the rate. So whereas the rate here goes a little bit gentler, with a high amount of tridoscamine, this protein aggregates very quickly. You can also see that here in this image. This is sort of an undisturbed A-beta-42 aggregation. It forms a small number of long fibrils. With the addition of tridoscamine, A-beta-42 forms a large number of small fibrils. The final way that oligomer kinetics can be used to address the toxicity of proteins is by targeting off-pathway species. So by using some sort of molecule to interact with the monomers and oligomers, it's possible to redirect their aggregation into a species that doesn't form an amyloid fibril, which takes secondary nucleation out of the mix for those aggregates. So a good example of that is resveratrol. This here is not a paper that I've done, but citations down here from 2010 that shows that resveratrol can interact with oligomers and direct them towards these large globular aggregates, which are not amyloid fibrils and are too large to affect the cells. This scale bar here is 1 micrometer. So they eventually become very large. This is similar to some of the things that your body does in the protein homeostasis system, wherein different proteins like molecular chaperones, heat shock proteins, interact with misfolded proteins and cause them to aggregate off pathway into harmless, larger aggregates. So the next mechanism we're going to talk about is targeting the physicochemical properties of oligomers. So before talking about any specifics, it's important to talk about the size, hydrophobicity, toxicity relationship between oligomers and toxicity. So as you can see with this sort of concept figure, the more hydrophobic and the smaller an oligomer is, the more toxic it is, because that makes it more likely to interact deleteriously with the cell membrane. You can see here in this other concept figure that a less hydrophobic oligomer might sit on the cell's surface, whereas a more hydrophobic oligomer will embed itself in and cause that membrane damage that we are trying to deal with. So in this case, the size and the shape of the protein sort of determines its destiny. These treatments attempt to address that relationship in order to make the proteins less toxic. A good example of this is the work that we've done with rationally designed antibodies. So these designed antibodies here are able to affect the oligomer size by making them significantly larger. So this is A-beta 40 oligomers without designed antibodies, and this designed antibody that binds to residues 34 through 40 makes their size significantly larger. Excuse me. You can see here, this is borne out in more, less visual assays. So this here is an absorbance measurement that shows the size of the oligomers, and here we've got more absorbance, and this is a count measurement that's performed on it. And you can see in all of these instances, the designed antibodies make the protein aggregates significantly larger. That's also borne out here in this atomic force microscopy diagram. You can see the shift of this curve to the right makes it so that you can see that the oligomers are significantly larger. So we would expect this to have a negative effect on toxicity, make these aggregates less toxic. Unfortunately for our designed antibodies, they also affect oligomer hydrophobicity by increasing their hydrophobicity. So here you can see an ANS fluorescence assay. ANS is a molecule that binds to hydrophobic residues on the surface of protein aggregates. And so an increased ANS fluorescence reading means that the protein is more hydrophobic on average. So as you can see, adding these antibodies significantly increase the hydrophobicity. This here is sort of the top of that curve. So you can see it in bar graph form. And generally, we would expect that larger size and increased solvent exposed hydrophobicity would balance each other out, and we would see no net change in toxicity. This is borne out by the data that we have. So this here is an MTT assay. MTT is a molecule that is metabolized by cells, and when it's metabolized, it fluoresces purple. So the more MTT that's metabolized in a given time, the healthier a cell is. You can see here that generally with the addition of these antibodies, cells did not get significantly healthier or less healthy than when they were just treated with misfolded protein oligomers. The third mechanism of action that we've been looking at that I'd like to talk about is protecting the cell directly from oligomer interactions. So this is a sort of newer line of effort that we've been looking at over the past few years. A very good example of a membrane displacing molecule is tridoscamine. We talked about tridoscamine a little bit earlier as it aggregates protein. Well, it also has the ability to displace misfolded protein oligomers from the membranes of cells. So here you can see that in picture form. These cells are highlighted in red, and the A-beta-42 oligomers are highlighted in green. So in an untreated cell, of course, there are no oligomers, and when A-beta-42 oligomers are added, they bind to the cellular membrane. That's what's causing toxicity. That's what's causing cell death. Now, when they're incubated with tridoscamine, or when the cells are incubated with tridoscamine, the tridoscamine is removed and the A-beta-42 oligomers are added, so that's the pretreatment protocol. The oligomers are not able to bind to the surface of these cells. You can also see that graphed out here. This also does have a measurable effect on the health of cells. So these here are reactive oxygen species measurements, and they show sort of the damage to cells by these A-beta-42 oligomers. You can see that the oligomers are highly toxic to cells, and when tridoscamine is added to the mix, the oligomers are much less able to interact with cellular membranes and cause toxicity. That's also borne out in this MTTSA. As you can see, the addition of tridoscamine at different molar ratios is able to significantly improve cell health. Something else that's interesting is that tridoscamine is able to do this on other toxins as well. So A-beta-40 oligomers, which are another sort of subset of protein misfolded oligomers, are also displaced by tridoscamine. HIP-FN fragments, which are a derivative of a toxin produced by E. coli bacteria, are also displaced from the surface of cells by tridoscamine. The way that we think that this works, based on the research that we've done so far, is that tridoscamine is able to sort of get in on the oligomers, sweep them from the surface of the cells, and it's able to sit where oligomers would normally interact. It's also able to sit over damage that oligomers have made in cellular membranes. So you can see here in this concept figure, tridoscamine sort of acting as almost a patch, if you will, for the pores produced by protein misfolded oligomers. Finally, I'd like to spend a few minutes talking about the protein homeostasis system. This is not something that my group has done research into, but it is something that researchers in the field have been looking at. So I think it's important that we talk about it. So the protein homeostasis system is your body's protein misfolding response that it's had this entire time. Chances are the protein homeostasis system is working in your body right now to deal with some sort of misfolded protein. Generally, this is very effective. However, the protein homeostasis system does get older with age, which helps to explain why we see many protein misfolding diseases as people get older. So as your body's defenses against protein misfolding start to weaken, your body's more susceptible to these protein misfolding diseases. And there's been some work looking into ways to upregulate the production of things like heat shock proteins and molecular chaperones in order to make your body better at fighting off misfolded proteins into old age and sort of employ a more natural defense mechanism against these diseases. Finally, I'd like to talk about some of the work that we've done over the past couple months on the role of membrane lipid composition and resiliency to protein misfolded oligomers and folded biotoxins. So, as we've discussed before in the amyloid hypothesis slide, there's significant evidence that protein misfolded oligomers act through a generic mechanism of toxicity. So they bind with the cell membrane as well as cellular receptors. They'll pretty much interact with anything. What this means is that to some extent, the cellular membrane is having an effect on these misfolded protein oligomers. And what we wanted to do is see whether that effect had anything to do with the exact composition of the cellular membrane. So some previous work in the field has implicated gangliosides and lipid rafts, which are domains on the cellular membrane that have higher densities of molecules, so lipids that are more closely packed together. They have been implicated in sort of being more susceptible to oligomer toxicity. In order to study whether that was the case and for what molecules that was the case, we employed a toxic biomolecule, melittin, which is the toxin in honeybee venom. We use this as a model for acute cellular toxicity that we think will be useful in understanding how biotoxins interact with cellular membranes, and is also an interesting analog to how A-beta-42 oligomers interact with cell membranes. Melittin forms sort of a disordered polypeptide chain until it interacts with cellular membranes, at which point it forms an alpha helical structure, inserts itself within the cell membrane, and does the same thing as A-beta-42 oligomers. It disrupts membrane integrity and ion homeostasis and causes cell death. Now, melittin is a much more severe toxin than protein misfolded oligomers, but that, of course, is by design. So what we've done here is we chose a number of membrane lipids that are implicated one way or another in Alzheimer's disease. And we incubated cells for 24 hours with different concentrations of these membrane lipids in order to change their membrane composition. Then we either left those cells alone, in the case of the blue line, that's the untreated control, or we treated them with 2 micromolar melittin in the case of this red line. So what these plots show is the health of these cells as measured by an MTT reduction assay relative to 1.0, which is the health of cells that weren't treated with any lipids and were also not treated with any toxins. Also, the melittin was left in for about 15 minutes because it's such an acute toxin. Normally, A-beta-42, when we use it as a toxin, we leave it in for about 24 hours. So one way of interpreting that data is what I've pulled up here. So these graphs show sort of the relative performance of cells under different lipid conditions within those lipid conditions. So each of these bars shows the health of cells treated with melittin at a certain lipid concentration relative to cells that were left untreated that also had that same lipid composition. So here you can see how the concentration of these key lipids affects the resiliency of the cells to melittin toxicity. So here in the case of sphingomyelin, which is present in lipid raft domains in higher concentrations than normal, we see that increasing concentrations of sphingomyelin make the cell less resilient to melittin toxicity. On the other hand, increasing concentrations of docosahexaenoic acid appear to make cells more resilient to melittin toxicity. In the case of icosapentaenoic acid and ceramide, we did not observe any linear effects. All right, so now on to some summary and some concluding thoughts. So in summary, there are a few different lines of effort for addressing the cytotoxicity of misfolded protein oligomers. We see in the literature today those 4 distinct methods that I've talked about. So by either modulating the kinetics of oligomer assembly, modulating the physicochemical properties of oligomers, protecting the cell membrane, or making use of the protein homeostasis system, it is possible to reduce the toxicity of misfolded protein oligomers. That being said, there are advantages and disadvantages to each of these mechanisms. So, for example, in the case that you are employing an acceleration mechanism that accelerates the rate of aggregation for misfolded proteins, you may be producing aggregates at a faster rate and reducing the amount of oligomers in solution at one point, but because fibrils are able to perform secondary nucleation, the more fibrils that are in solution, the more thermodynamically favored aggregation is, the more favored this protein misfolding continues to be over time. Additionally, because it is possible for oligomers to break off of fibrils, it's possible that you're creating almost reservoirs of oligomers that could at any time break off and interact with cell membranes further. Now, fibrils are generally stable, so that's not a particularly likely event for any individual fibril, but it's important to note that there are risks associated with that. The same is true for all these other methods of addressing toxicity. What this also highlights is that there may be a shot at doubling up on these lines of effort. So a potential drug for a protein misfolding disease might be able to make use of an ingredient, a therapeutic that uses one line of effort and another that uses another line of effort. Because these therapeutics might act by different mechanisms, it might be possible for scientists to create a drug that deals with multiple steps of the aggregation cascade and the ensuing toxicity at once, which may be more effective than just employing one method at once. This also highlights that there's sort of more opportunities for a rational approach to addressing toxicity by understanding how a molecule interacts with the whole process of protein misfolding toxicity. It's possible to more easily understand what exactly we should be studying to see how effective that molecule is. And hopefully that will eventually lead to a real therapeutic that passes 3 stages of clinical trials and is able to be used in patients in the future. So for future directions, for the field, we hope that our work and the review that I helped write last semester helps in understanding rational drug discovery techniques for protein misfolding diseases. We also hope that our work with design antibodies helps in diagnosing protein misfolding diseases. So most protein misfolding diseases aren't diagnosed until they're in very late stages. For instance, in Alzheimer's disease, most people don't screen for Alzheimer's regularly. They sort of find out they have it when they start to experience cognitive symptoms. Now, that, of course, is not ideal for treating these diseases because if they're already in an advanced stage, there are already amyloid fibrils sort of dispersed throughout the brain. They're already causing severe damage to neurons, and it's more difficult to treat a disease at that stage. So having rationally designed antibodies as molecules that can selectively bind to parts of these proteins means that we may be able in the future to have a reliable test early on for whether someone has the aggregation of misfolded protein that could turn into Alzheimer's disease or Parkinson's disease or any of these other protein misfolding diseases. For our group in specific, one of the things we'd like to do is apply the research that we've done on membrane lipids to protein misfolded oligomers. So right now we suppose that the mechanism of action is similar enough that membrane composition ought to have an impact on a cell's resilience to misfolded protein oligomers as well as folded biotoxins. However, we are currently in the process of testing that. I actually have to run a step on one of those experiments right after this talk is done. Additionally, we'd like to analyze new molecules for potential sort of capability as therapeutics. So a good example here is this molecule, the name of which I cannot share with you at present. As you can see, the addition of this molecule is able to significantly reduce the toxicity of A beta 42 oligomers. And we think that the effects of this molecule might be interesting and definitely warrant further study. Finally, we'd like to take the research that we've done on A beta 42 and the research that the field has done on A beta 42 and apply it to new protein targets. Specifically, we're looking at tau protein to see how these countermeasures affect tau aggregation. And we're interested in knowing sort of just how generalizable protein misfolding disease research is, because we know that the process of primary and secondary nucleation, the aggregation cascade, the amyloid hypothesis, these are common threads that underpin protein misfolding diseases. But to what extent do these all stay the same? Some molecules may have different effects on different types of protein. Trodoscamine, for instance, accelerates the aggregation of A beta 42, but decreases the rate of aggregation for alpha-synuclein, which is the protein implicated in Parkinson's disease. What are these effects and how do they work across the entire field? This is something that we'd like to study in more detail in the future. All right, at this point, I'd like to thank all the people that made this talk possible. So Dr. Limbacher, thank you for being an excellent PI and really bringing me into the field and showing me the ropes. I definitely wouldn't be where I am without you. Colonel Wynn, thank you so much for putting this on, for giving me this opportunity. And thank you to all of you for giving me this opportunity. This is really something special. I never in my wildest dreams expected I'd be given a talk like this as an undergrad. I'd also like to thank Colonel Wynn for being my department academic counselor for the past 2.5, almost 3 years now. Goodness knows I've put her through enough with my schedule. I'd also like to thank the other instructors in my group, Major Hollows, Major Lafort, and Captain Wynn, and the other cadets in my research group. For funding sources, I'd like to thank the Army Research Laboratory, the Defense Threat Reduction Agency, the Stamps Family Charitable Foundation, and the Department of Chemistry and Life Science. At this point, I'd like to open the floor for questions. I'm happy to go over any part of the presentation or talk about any sort of further questions you may have.
If you'd like to ask a question, you can unmute your microphone. That would be fine.
Yes, I have a question.
Yes, ma'am.
The misfolded proteins are made in the cytoplasm, correct?
Yes, that's right.
And yet your diagrams seem to show that the effects are on the outside of the cell, on the outer membrane. I don't understand. Are you saying that these misfolded proteins are extruded and then affect the outer membrane? Or is it just that they're going through from the inside?
It's not something that I've spent a lot of time studying myself, but of course, some of the misfolded proteins do make it outside. So some of them, it really depends on the protein. Some of them may be marked for secretion. Protein misfolding, it covers a lot of different possible peptide chains. So some of them may get secreted intentionally. Some of them may end up unintentionally on the outside of the cell due to their interaction with the cellular membrane. In any case, once they've made it outside, they begin to do some serious damage. And as you mentioned with that figure, there are also intracellular effects with neurofibrillary tangles within the cells in Alzheimer's disease.
Yeah, I would have expected that. I was looking to see what was going on inside the cell where these are actually being made. And I wouldn't be surprised if you had more effects from the inside than from the outside, but that's just my gut reaction.
Yeah, so Ryan hit it spot on. Beta amyloid, the protein involved in Alzheimer's, is secreted. So the way it misbehaves is all extracellular versus tau
Ah.
that's intracellular.
Uh-huh. Okay.
Good answer, right? So Ryan, which strategy do you think is the best?
PI is asking me, what is your favorite protein question? So, I mean, me personally, I'm really interested to see what we have going on with displacing oligomers from the surface of cellular membranes. To me, at least, it seems like addressing the sort of thermodynamics is more difficult, especially at the stage that people find protein misfolding diseases. At this point, the aggregation cascade's already been going on for, I mean, years. So being able to protect cells by sort of competing those binding environments, that to me seems like a good way forward and a way that can address the toxicity of these misfolded aggregates at later stages in their in the disease's progression. But of course, at this point, there hasn't been any therapeutic that's made it past clinical trials. So it's really anybody's guess as to what the most effective one is. And I'm sure we'll all be very excited when we find out which one it is.
So Ryan, I have a question for you.
Yes, ma'am.
So you've mentioned that with Alzheimer's and many similar diseases that the symptoms really don't start showing until the disease itself has progressed far enough. And so by that point, it's kind of hard to treat it. But what, and I, of course, I don't know, my background is physics and space. So I'm not really sure of the current status on the genetics associated with Alzheimer's and similar diseases. But what if you could test for the genetics and then early treat some of those things? Or is there a way, one, to test for the genetics, and two, would some of the options that you highlighted here be able to treat early to then be a preventative measure rather than a reactive measure?
That's a good question, ma'am. So it's sort of yes and no. So there's not exactly an Alzheimer's gene, so to speak. There's not sort of a set of genes that determine whether the protein you produce or is going to misfold, or that your protein homeostasis system is going to weaken to such a point where it can't contain it. There's a lot of factors at play. However, there are, in the case of some amyloidogenic diseases, there's some genetic predisposition. So Huntington's disease, for instance, is a genetic disorder, and that's a protein misfolding disease. It may be possible to determine whether someone's producing proteins that are slightly more likely to aggregate, but it's difficult to have a genetic test directly, so to speak. So that's why we're interested in the effects of these design antibodies and the ability to detect protein aggregation at the early stages, because while it may be difficult to say outright who is going to get a misfolding disease, it might be easier to determine who is beginning to get a misfolding disease, say 5 years before symptoms start showing, rather than 2 years after.
Awesome, thank you.
Excuse me, for Alzheimer's disease, there is not yet any method to test if you can have possibilities to have Alzheimer's disease, no? It's only cognitive tests, that's what they're doing, or already they have an antibody test that they can do.
Oh, that must be a gap in my knowledge. I would love to hear more about it.
There is no, it's only cognitive tests at this time for Alzheimer's disease.
So there are some radioligands that combine to the plaques, and so these you can quantify with some PET imaging, but the main issue there is that you don't really do that assay until someone's had protein aggregation, often for a decade.
Okay.
So I mean, I guess in theory you could do these to healthy people, but the risks associated with putting the radioligands in you are not worth the process itself.
Okay.
Yeah.
Okay, so I guess that's where my question stemmed from. So if your family had a family history of the genetic or of the disease itself, then theoretically there would be a genetic connection. So maybe there would be an opportunity to pre-treat.
Yeah, chronologs, you're exactly right. But unfortunately, the familial forms of Alzheimer's only account for about 5% of the cases. So about 95% are
Okay.
sporadic with no familial genetic association.
Okay.
So in those populations where it's the 5%, you can do the gene sequencing and understand. But again, that's usually not something people find until families have histories of Alzheimer's disease.
I understand. Very similar to cancer, I guess, is they're starting to do a lot of genetic testing with cancer as well.
Exactly. So there's definitely, as much as we try to do therapeutics, it's equally as active in the field for better diagnostic assays. So we can actually, once we have a good therapeutic, know when to give it to people. Even as, I mean, in theory, you could have statins like you would for cholesterol drugs with neurodegenerative disease, but there's surely side effects associated with even the most bioavailable molecules that we've studied. So it's definitely a battle on both arms. There's one antibody in clinical trials, aducanumab, that's gotten a lot of attention because it failed clinical trials and then they brought it back and now it seems like it's failing again. But there was another one, a phase 2 trial just commenced last week, and it looks really promising. So some of these antibody therapeutics are great, not just for diagnostics, but they also seem to be able to clear the plaques from the brain. So a lot of different research going on all these arms that Ryan described really well to try to find something that can help us treat patients.
Awesome. Thank you all. And by the way, I just wanted to say, Ryan, that was a fantastic presentation. So thank you for inviting me.
Thanks, ma'am. I really appreciate you taking the time to come. I love your background too.
I guess I was just thinking of one thing. When you showed the chaperone proteins, has there been research on those particular proteins that are involved and what they are basically at fault in terms of not doing their job per se?
Yes, ma'am. There's been a lot of work actually on understanding what is going wrong with the protein homeostasis system that's enabling these misfolding proteins to aggregate. The answer is a little bit trickier than one specific thing that's going wrong because there's so many different components of the protein homeostasis system. There's so many different chaperones. There's so many different regulators. It's sort of a lot of things going wrong at the same time. There's a lot of things going wrong. So, well, it's difficult to say, like, say, a secretase inhibitor being downregulated is the sole cause. There's definitely a number of individual steps that researchers are looking at. I regret that that's a gap in my knowledge specifically, that it's not something that we really study in depth in our group, but it's definitely something that people are looking at to understand if there's one, fulcrum point at which the aggregation cascade is able to happen.
Right. Thank you. Are there any other questions?
I have a question. So Ryan, I know you're focused on amyloid beta 42, but comparing that to some of the amyloids that are more functional. So for instance, different hormones that can be secreted are typically stored as a amyloid type phase. And obviously these are not quite as toxic. Have you done much research into looking into those to kind of juxtapose that against something that's obviously more cytotoxic?
Well, sir, I haven't done any research on that specifically, but there's a pretty interesting paper if you want to read it. It's a review from 2006 called, I I think it's protein misfolding, functional amyloid, and human disease. If you look up Dobson-Keaty Annual Review 2006, that should be the right paper. It talks about sort of the ability of many different proteins to aggregate into an amyloid state, and how there's, as you said, there's examples of organisms using functional amyloids to sort of perform a task that a single protein might not be able to do. So protein aggregation isn't always something going awry. And it's partially that ability of proteins to aggregate that causes these proteins to not reach their native state and end up aggregating. Because the ability of proteins to misfold and aggregate isn't reserved to just these specific proteins. It's sort of a possibility for any protein that's sort of on that edge of stability, where it's just a little bit more stable to reach the native state is to reach one of these off pathway states. So you'll see some of the same things going on with functional amyloids as you'll see with dysfunctional amyloids, just with different effects, where a functional amyloid is performing a job that the body wants, and a dysfunctional one is essentially producing a lot of oligomers and running itself wild to produce so many oligomers that it eventually starts damaging your body.
Thank you.
Yeah, Daniel, that's an awesome question. So a lot of people are using functional amyloids for material science as well. So there's water purification units based on amyloids. Recent science paper showed it might be implicit in Drosophila forming memory. So I mean, it's very diverse. The challenge is just getting sufficient amounts of protein to go after these functional amyloid studies. So from a material science perspective, you need grams of this stuff, and they can be hard to synthesize or produce. But definitely really important parts to look at, things we're interested in too.
All right, so Ryan, I have a question for you. So great talk this evening. You've presented a lot of different areas of research and different pathways that you could take the project, and you have some current work going on right now. So what's your immediate, what's your short-term study in terms of what are you trying to, like, what's your near-term question that you're trying to answer that will advance this holistic research effort?
Sure, ma'am. So what we're studying right now, at least my sort of area of interest that I'm devoting most of my thought new experiments, is how the work on membrane lipid composition applies to misfolded protein oligomers. So I've got to apply some toxins tonight, and this will be the first round of experiments where we've got some cells that we change the composition by doping in sphingomyelin and docosahexaenoic acid in two different types of cells. And we're looking to see whether those same effects that we saw with melatonin hold true with DHA and sphingomyelin with A-beta 42. Right now, a lot of what we're doing in the group is we're looking to publish some stuff with that undisclosed molecule that I mentioned earlier. We've got a lot of data on that, and we think it's going to be pretty big for the field when it comes out, because the molecule in question has some advantages over some other molecules that do similar things that make it worth further study. So sort of on those two arms with publishing and doing further experiments, we're hoping to show what we've got that's pretty strong and expand some of our findings to the question of protein misfolding.
All right, awesome. Looking forward to seeing your next next presentation or next paper. Thanks.
Any other questions?
All right. Well, if there are no further questions, I'd like to thank everybody again for their time. I had a great time presenting,
Yeah.
good time listening, and I hope I was able to sort of provide some insight into the field, as it were. I'll stop sharing now.
Isn't that the point where you plug your YouTube channel now, Methanol Lab?
Oh, boy. I'm not going to hear the end of this, am I?
You
Can stop
started.
the recording. I hope that's okay. Okay.